Hi, everyone. Welcome to Foresight's Molecular Machines Group. I'm so excited to have James Seale here today with us. You have not only been a Foresight Fellow, but you're also working and have worked with and collaborated with Fraser Stoddart, who a long time ago won our Feynman Prize for his work on molecular machines. And then a few years later afterwards got co-awarded the Nobel Prize for his work. And so I'm really excited to have you share a little bit more about your work here. There's also shorter presentations online already. One of them that you gave at a Foresight workshop a few years ago. So there's quite a bit of Foresight content on you out there now, but we're dying to hear a little bit more about what you've been working on recently. And yeah, please take it away. I'll share a lot more info about you here in the chat. But for now, I'm excited to have you on to discuss artificial molecular pumps operating on polymeric micelles. Thank you so much for coming. And the stage is yours.
Yeah, thank you for having me and for the kind introduction, Alison. I am a final year graduate student in the Stoddart group, Richard Stoddart's research group. I'm at Northwestern University, and I'm very excited to present my talk, Artificial Molecular Pumps Operating on Polymeric Micelles, that I've been working on for the last 2 or 3 years. There we go. So before I talk about artificial molecular pumps, I want to talk about nature's molecular pumps. One example of these is the sodium-potassium transmembrane pump, which is found in almost all animal cells. So a really ubiquitous piece of nature's nanotechnology. These transmembrane pumps are found embedded in cell membranes, as shown in this image here. And they will hydrolyze ATP, and using the energy released, export 3 sodium ions from inside the cells to the outside, and then import 2 potassium ions for every cycle. So the overall effect of this pump is to create concentration gradients of sodium and potassium in opposite directions, and then also to generate a transmembrane potential because 3 cations are exported and only 2 are imported. And nature's molecular pumps are extremely complex and also very diverse protein-based machines. Here's a crystal structure of the sodium-potassium ATPase, which is just another term for the pump. You can see that there are multiple different parts of this machine. It weighs 165 kilodaltons, so it's very large. I think it's just popped up again. Hide it. There we go. But there are a huge variety of these molecular pumps found all throughout different organisms. So halorhodopsin is a chloride pump that belongs to Archaea. As you can see from this crystal structure, it is much smaller than the sodium-potassium pump. It consists of just a single subunit. And rather than using hydrolysis of ATP to drive its pumping action, it uses the absorption of light by a chromophore that is embedded in the transmembrane domains. And so here there's a little bit of diversity in the kind of ion that is pumped. The sodium-potassium pump transports cations in both directions, while halorhodopsin pumps chloride ions, so anions, into cells. There are also molecular pumps that will transport molecules, for example, glucose pumps. And in bacteria, there are the famous drug export pumps, multi-drug efflux pumps. Those can transport antibiotic molecules from inside the bacteria to the outside, which is one mechanism by which bacteria can become resistant to antibiotics. So given how important natural molecular pumps are for, all organisms, it has been interesting to researchers to try and create artificial mimics of those pumps. And one class that has become popular over the past decade are what I will call ring-threading molecular pumps. So a variety of these have been reported in the past decade. Exactly what you define as a pump is not set in stone, but I would say that there are 6 different kinds of pumps, ring-threading pumps, that have been reported in the last decade. At the top of this slide is one example from the Lee group that was reported in 2021. This molecular pump is capable of transporting these red macrocycles, which are crown ether rings. And it does this by capturing them when it forms this bond here. This is basically the pumping part of the molecule shown on the left side. And then the rings are transported onto this collecting chain, onto the triazole units of the collecting chain. So this pump has already pumped 2 rings and has captured another in its binding site, as shown on the slide. In 2022, there was a circular molecular pump reported by Coulomb-Ju's group. This also operates with a kind of crown ether. It's more like a pseudo crown ether, and it is able to clip this crown ether on the binding site of the pump, which is the blue part of the molecule, and then pump them onto this collecting chain, which is actually attached again to the pump head. Hide the main controls.
Just moves yes, perfect.
Yeah. So you can see from these two examples of artificial ring threading pumps that they are structurally very different to nature's molecular pumps, some examples of which I showed in the last slides. But although they are structurally very different, they are conceptually pretty analogous in that both the natural and the artificial molecular machines are capable of the active transport of solutes in solution. So to outline the similarities and differences between these systems, I just want to draw your attention to this diagram at the bottom right of the slide. In the top panel, we have two systems that are at equilibrium. Here, there are cell membranes and a distribution of ions, which are these yellow dots. And at equilibrium, there is an equal concentration of ions inside the cells and outside the cells. When you attach these blue molecular pumps to the membranes and then selectively pump ions inside the cell, you end up with what they call a concentration gradient. That is high concentration of the ions inside the cell as compared to outside the cell. Similarly, if you have a mixture of thread molecules and macrocycles in solution, assuming there is no natural affinity between the macrocycles and the threads, which is true for the vast majority of pairs, then you'll end up with basically a vanishingly small amount of any of those macrocycles threaded by the linear molecules. When you attach molecular pumps to one or both ends of the threads and then pump rings onto the macrocycle, you end up storing the rings at high concentration in this kind of threaded compartment. Both natural and artificial molecular pumps are capable of generating non-equilibrium systems. That's the key message. The ring threading molecular pump that I'm going to talk about today was developed by the Stoddart group in 2017, so way before I joined the group, and it was inspired by previous work going back for years before that report. At the top of the slide, I'm showing the structure both of the pump, which is this linear molecule here, and its cargo, which is shown on the left. Cyclobis paraquat p-phenylene macrocycles, I will just call them CBPQT. So you take, when you operate the pump, you take the macrocycles and the pump, you dissolve them in solution, and then you reduce the system. And when you reduce the system sufficiently, you will form this host-guest complex, which is shown in purple in the second panel. This is formed by the radical pairing interactions between the Viologen unit on the pump and the Viologen units of the CBPQT rings. When the system is then oxidized back to its ground state, the radical pairing interactions that made the host-guest complex are destroyed. And in their place, there is a huge coulombic repulsion between the molecular pump, these positive ions shown in the structure here, and the positive charge of the rings, which are tetracationic salts. Because of this repulsion, the ring is forcefully ejected from the binding site, and because of the asymmetric design of the molecule, it will move preferentially to the right, as shown on the slide, and it'll end up interlocked on the collecting chain, as shown in this bottom diagram. I just want to compare and contrast natural and artificial molecular pumps a little more. First, one of the main differences, I think, is that ring threading molecular pumps are very minimal. They have a low molecular weight. The molecular pump that I'm going to talk about in today's work weighs 860 Daltons. And you can compare that to halorhodopsin, which is actually one of nature's smaller molecular pumps, which weighs in at 26.9 kilodaltons. In that sense, ring threading molecular pumps are very atom efficient. They're able to demonstrate active transport using fewer atoms than transmembrane pumps or natural transmembrane pumps. The second feature, I think, that jumps out from these ring threading pumps is their abiotic mechanism of active transport. So these molecular pumps transport macrocycles by threading them and then store them threaded on a collecting chain. And this is not like anything that you see in natural molecular pumps. In light of those two pretty big structural differences between ring threading pumps and natural pumps, it's been interesting to see what kind of scenarios these ring threading pumps can be operated in. So they've been operated as free small molecules in solution since the earliest reports around 2015. They have been attached to the termini of polymer chains for the synthesis of precise and very ring-dense polyrotaxanes since 2020, the earliest report. And they've also been attached to solid surfaces where the artificial pumps can then transport the rings from free solution to the solid state. And that's since around 2021. But ring threading molecular pumps have not yet been tested operating in aqueous supramolecular assemblies in imitation of their biological counterparts. And so just to return to the example of the sodium-potassium pump and halorhodopsin, this time I'm showing them in their natural environment, which is embedded in the cell membrane. And so that was the goal of this project of my PhD, was to try and integrate ring threading molecular pumps into aqueous supramolecular assemblies and test their capacity to transport rings. And so this is a diagram outlining what I was going for. I settled on polymeric micelles as the aqueous supramolecular assembly that I wanted to study, and I'll explain a bit why I chose that system or that platform in the next couple of slides. And so some polymeric micelles are shown by these gray threads, which have self-assembled in aqueous solution to form these spherical molecules, or spherical aggregates rather. And you can see that these are colored molecules. These are the artificial molecular pumps that are attached to the termini of the polymer chains and therefore sit on the corona, on the edge of the micelle particles. The cargo of the molecular pump, the CVPQT rings, are shown by these blue rectangles, which are floating in free solution. So since CVPQT is a tetracationic molecule, it can form a variety of different salts. And in this study, I'm using it as its chloride salt, which is extremely hydrophilic and which is expected to be distributed in aqueous solution rather than to associate with the hydrophobic micelles. And so under equilibrium conditions, we expect this kind of distribution of the components of this solution. I then wanted to see whether by operating the molecular pumps, the hydrophilic rings could be transported from free solution to the interior of the polymeric micelles. So this project was based on some prior work from the Stoddart group that was looking at the compatibility of these molecular pumps with polyether chains. And so it was shown that by attaching molecular pumps to the termini, either of PEG chains or PPG, polypropylene glycol chains, you could form precise and stable rotaxanes. And so we knew that this redox-driven pump was compatible with polyethers. And as it turns out, there is a triblock copolymer of polyethers called pluronic, which is known to form micelles in aqueous solution. Here is the structure, the generic structure of a pluronic thread. The PPG segment is in the center, and it's flanked on either side by polyethylene glycol segments. These pluronic molecules have been studied for a long time, especially in light of their micelle-forming tendency, which has been known for over half a century. So the driving force for formation of these pluronics, by the way, is the fact that the polypropylene glycol segment of the chains is very hydrophobic. And so in aqueous solution, there is a tendency for these PPG segments to be buried inside the micellar core, where they don't interact with water molecules. And instead, the PEG chains, that are the termini of the whole chains, form the corona of the micelles, and those are a little more hydrophilic, and so it's okay for those to interact with water molecules. So I wanted to synthesize pluronic threads with the molecular pump on either end. And this is the procedure that I used to do that. I started with a commercial sample of pluronic, shown at the top left of the slide. I reacted that with methane sulfonyl chloride to produce this pleuronic bismethylate chain. I then reacted that with sodium azide, which by an SN2 reaction can form pleuronic bisazide. The bisazide can then be reacted with the standard molecular pump design, which, as I mentioned, was first reported in 2017, using a click reaction in aqueous solvents to produce this, what I will call the pleuronic bis pump molecules, or pleuronic BP, in a pretty decent yield, 77%. These pleuronic BP chains were isolated by reverse phase column chromatography, and a number average molecular weight for the entire chain, including the pumps, of about 9,000 grams per mole was calculated. So proton NMR provided the evidence that I had synthesized these molecules and also allowed me to calculate that number average molecular weight. So this is the proton NMR spectrum. It is quite complicated because there are a lot of different parts to these molecules, but I'll just draw your attention to some of the key parts. So there are some very large, broad resonances. For example, here at 1.2 ppm, this resonance corresponds to the methyl protons on the PPG segments. At around 3.6 ppm, there are a bunch of broad and overlapping resonances. These similarly correspond to the protons on the PEG chains and some other protons on the PPG chains. I've made a magnification of the aromatic part of the spectrum because that contains the resonances corresponding to protons on the molecular pumps themselves. And that is also where the proton resonances of the cargo of the pump, the CBPQT rings, will appear later on. So it's good to pay attention to this part of the spectrum. Having synthesized those pleuronic bis pump molecules, I wanted to test whether they would form micelles, and I found that they did. Specifically, I ended up using this kind of solution for the experiments in the study. So that's pleuronic bis pump molecules at a concentration of 5 mg per mil and in deuterium oxide rather than normal water. The reason I used deuterium oxide was to allow proton NMR and other NMR spectroscopies both before and after attempting pumping. So these micelles were formed in the presence of the cargo of the pump, the CBPQT rings, which is of course important for later pumping studies. And the solution of these constituents, I'm going to start calling the pre-pump micellar solution, just for simplicity. Dynamic light scattering and zeta potential results provided the first pieces of evidence that I was forming aggregates in solution using these polymer chains. So the DLS results are shown here in this bar chart, and I calculate a mean aggregate size of 11.6 nanometers. The zeta potential is quite high and positive, and this is what you would expect for micelles that display the positively charged molecular pumps on their surfaces. Just for comparison, if you form micelles of just plain pleuronic without any pumps attached to it, the zeta potential of those micelles is around -1 millivolts. And now forming the micelles of pleuronic bis pump, the zeta potential is more like positive 43.8 millivolts. So DLS and zeta potential provide evidence that there are aggregates in solution, but they don't confirm that those aggregates are micellar, nor that they're spherical. So it can't really tell us about the shape of the aggregates. And to check out the morphology, I use transmission electron microscopy to take images of the micelles directly. So this was done by two methods. In the dry state, by taking the micellar pre-pump solution, dropping it onto a substrate, drying that substrate, and then dropping a uranyl acetate stain onto the substrate, which provides a better contrast for the TEM images. And this picture here shows micelles of diameters in a range 10 to 15 nanometers, which is in good agreement with what I saw by DLS. And this image was taken by Dr. Tyler James, so I'm very grateful for his expertise and help with that. Similarly, cryo-TEM, first I'll explain what that is. So cryo-TEM is where you first cryogenically freeze your solution rather than dropping it on a substrate and drying it. and then image that frozen solution directly. And so cryo-TEM generally gives a better or more reliable insight into what's happening in solution without any drying artifacts. And the cryo-TEM results show that micelles, roughly spherical micelles, are present in solution. Here, the diameters of these particles are lower, 5 to 7 nanometers. I would note that it's actually typical for cryo-TEM to register low diameters for polymeric micelles as compared to other methods like dry TEM and DLS. And this cryo-TEM image was taken by Dr. Eric Roth, so grateful to him as well. So now I had made these micelles in which the molecular pumps were integrated, and I wanted to figure out whether I could successfully operate the pump on these aggregates. I started with the pre-pumped micellar solution as described, and I loaded a couple of milliliters of that into a nitrogen-filled purge box. The reason I do this is that the molecular pumps here, they operate through redox stimuli. So first reducing the solution and then oxidizing it. And in order to get efficient reduction, I want to make sure that there is as little oxygen as possible in those solutions. That's why I put it in a nitrogen purge box. I reduced the solution with zinc dust. So this is a heterogeneous reducing agent. I would just drop some of the zinc powder into the solution directly and then stir for 30 minutes, and the solution would turn deep metallic purple color, which is associated with the first stage of operation of the molecular pumps. The reduced solution was then removed from the purge box and then transferred to a round bottom flask, where I then bubbled air through the solution. And so here the air is acting as the oxidant. And what happened was that the deep purple metallic color was gradually lost from the solution and replaced by a pale purple or pale blue color, by which I knew that the oxidation had gone almost or entirely to completion. And it took about 10 minutes to do that oxidation. Then the post-pump solution, which is what I'm going to call the solution after performing this redox cycle, that could be analyzed directly by NMR spectroscopies, DLS and zeta potential again, and also transmission electron microscopies. The proton NMR of the post-pump solution showed resonances that are characteristic of mechanically interlocked CBPQT rings. And so this indicated to me that the operation of the pumps had been successful. So here I have a couple of pre-complicated NMR spectra. So at the top of the slide, I'm showing the constituents of the solution. We have the fluoronic bis pump molecules shown here with some assignments. There are the free CPPQT rings. So those are the ones that are still free in solution and not associated with the micelles. But then there are also these interlocked CPPQT rings. Here I've just colored them magenta to distinguish them and to color code these NMR spectra. And once the rings are pumped onto the polymer chains, we would also expect that they would be integrated into the micelles, since the micelles are composed of those polymer chains. So there are two NMR spectra here. The top spectrum is the pre-pump micellar solution, and there are 4 key peaks here. These peaks highlighted blue. Those are the 4 unique protons on the CBPQT rings. After the pumping cycle, there are 4 new peaks that have appeared in the spectrum. They are colored purple or magenta, and those are the resonances corresponding to interlocked CBPQT rings that have been pumped from free solution to micelles. Let me just remove this mutant control. Okay. And so by measuring the integrals of these new magenta peaks, I can calculate the pumping efficiency of the system. And here I'm defining the pumping efficiency as the number of pumped rings divided by the number of molecular pumps present in the system. And what I calculate is a pumping efficiency of 65%, which means that 65% of these individual molecular pumping cassettes have successfully transported a CBPQT ring. Another kind of spectroscopy, that's the NMR or diffusion ordered spectroscopy, provides some useful evidence that the macrocycles have been transported from free solution to the micelles. And so what DOSE NMR can tell you is the diffusion rate associated with the resonances present in your NMR spectrum. And here's the dose NMR spectrum for the pre-pump solution. So this is before I attempt any pumping. And you can see that there are two key peaks on this 2D spectrum shown on this axis here. This corresponds to the free CPPQT rings, which are diffusing very quickly. So we can measure this diffusion rate from the dose NMR results. And you would expect that these small molecule rings in free solution would be diffusing very quick relative to the micelles. So the micellar diffusion rates, this span a range from 7.2 to 3.2 times 10 to the minus 7 centimeters squared per second, and they're shown down here. And so this tells me that the rings do not associate with the micelles under equilibrium conditions prior to pumping. Next, I'm going to show the dose NMR spectrum that I took of the post-pump solution. And this indicated still that there are the free CPPQT rings present in solution. That's because I used an excess of the CPPQT rings. So, not all of them are pumped into the micelles. But also there's evidence that those rings that have been pumped and are now mechanically interlocked on the polymer chains are diffusing at the same rate as the micelles. And so to really show that more clearly, I'm going to zoom into this aromatic region of the spectrum that's highlighted by this box. So here you can see the free CPPQ2 rings. Their resonances are up the top. They're diffusing quickly, but the new magenta resonances of the interlocked blue box are diffusing at the same rate as the micelles, specifically at 7.56 times 10 to the minus 7 centimeters squared per second. And that constitutes a reduction of 80% in their diffusion rate because of the fact that they've been pumped into the micelles. Next, more transmission electron microscopy was performed just to check that the system was still micellar after the operations of the pump. And I found that it was, both by dry TEM and again cryo TEM. I can see the presence of spherical micelles of roughly the same dimensions, maybe a little bit larger by the dry TEM results. Again, these images taken by Tyler and Eric. The DLS and zeta potential of the post-pump solution showed a slight increase in the diameter and zeta potentials after pumping. Here again are the DLS and zeta potential for the pre-pump solution. And just below them are going to appear those for the post-pump solution. Now the average micellar diameter is 13.8 nanometers, slightly higher than the 11.6 nanometers prior to pumping. Also, the zeta potential has increased from an average of 43.8 millivolts to 54.7 millivolts. And both those changes are what we would expect when you're importing macrocycled, positively charged macrocycles from free solution to the micelles. And I was satisfied that we had seen pumping from free solution to the micelles, but I also wanted to investigate the effect that these micelles had on the pumping efficiency of the ring-threading molecular pumps. And so to that end, I performed a non-micellar control experiment. And it began with this molecule shown in the top here. I'm going to call this oligoethylene glycol bis pump. So rather than a polymer chain, these molecular pumps are attached to a small oligomer that is a hexaethylene glycol chain. And so I tested the operation of these small molecule bis pumps under equivalent conditions to those performed on the micellar pumps. So same concentration of pumps and rings, D2O solution, and again, the same zinc, dust, and then air sparge redox procedure. And by NMR, I can see that I form two major constituents in the post-pump solution. The major of those two is the 3-rotaxane, that is counterintuitively the rotaxane with 2 rings on it, shown at the top here. And then the minor product is the rotaxane with just 1 ring, so where only 1 pump has successfully transported a ring onto the chain. And from the proton NMR, I was able to calculate a small molecule pumping efficiency of 91%, a little bit larger than the 65% that I saw for the micellar pumps. And again, proton NMR was really useful for calculating this pumping efficiency. Here I have the pre-pump solution for the small molecule control experiment shown in the top. All these red peaks correspond to the molecular pump and their chains, and then the blue peaks again to the free macrocycles. The magenta peaks correspond to the 3-rotaxane, that's with 2 rings, and the green peaks to the 2-rotaxane, that's with 1 ring. And so by comparing the integrals of the magenta and green species, I can calculate the amount of the 3 and 2-rotaxane that are in solution. And from that, I can infer the pumping efficiency. One thing I should point out is that there is no detectable amount of the oligo, e.g. bis pump molecules present in the solution after pumping. So virtually all of those molecules have been transformed into rotaxanes. Finally, I did some UV-Vis spectrophotometry to try and analyze the difference between the micellar pumping and the non-micellar pumping. The spectrum for the micellar pumping is shown on the left, and that for the non-micellar pumping is shown on the right. And what these results indicate is that there are some competitive vialogen-to-vialogen interactions that occur in the micellar pumping scenario, but not in the non-micellar pumping scenario. And I attribute the slightly lower efficiency of the micellar pumping to the presence of these competitive supramolecular interactions. So just to make a little more sense of these spectra for people unfamiliar, the tris radical tricationic complex, that is basically the host gas complex, is the first stage of pumping with this redox-driven pump. That has a characteristic absorption in UV-Vis of around 1100 nanometers. And so that is observed pretty clearly, actually, in both the spectra. Here in the non-micellar pumping case, it's very distinct, but here in the micellar pumping, it's actually buried as a shoulder of this larger peak attributed to competitive binding. And so it's well reported in the literature that pi dimer interactions or polymerization between vialogen units results in a UV-Vis absorption peak of around 900 nanometers, or at around 900 nanometers. And so that's how I can attribute this new peak to competitive vialogen interactions. And now I'll just summarize what I've gone over. Amphiphilic polymer chains can be used to drive the formation of supramolecular assemblies bearing ring-threaded molecular pumps. The active transport of hydrophilic small molecules from free solution into hydrophobic assemblies has been demonstrated. And it was found that this active transport occurs with a slightly lower pumping efficiency in the micellar scenario than in a small molecule control experiment. So next, I'd just like to acknowledge the many people and institutes that have made this research possible. So my collaborators on this project, of course, I'm very grateful to Fraser for giving me a home in his lab and for guiding me over the last 5 years, and also to the various collaborators that have made this research possible, some of whom I mentioned throughout. I'm also grateful to the Stoddart Group as a whole and to Northwestern University. IMCIRC is one of our facilities at Northwestern where I did a lot of NMR for this project. Also grateful to the International Institute for Nanotechnology for supporting this research, and of course to the Foresight Institute for supporting me and giving me the chance to speak today. But I know that you guys in Foresight like to talk about future goals for the field, so I added a couple extra slides giving my thoughts on that. So to my mind, research into artificial molecular pumps can be split into unnatural applications and biomimetic applications. So an example of an unnatural application would be the use of these ring threading pumps to make polyrotaxanes, which is really unlike anything that you see in nature. A biomimetic application is more considering or starting from the point of view of we have these artificial molecular machines. They are structurally very different to natural molecular machines, but can they operate under similar scenarios? And so that biomimetic kind of goal was the thinking behind this project. And my personal bias is towards the biomimetic applications, so trying to see if artificial molecular pumps can perform tasks similar to their natural forebears. And in particular, I think probably one of the major questions on that line is whether these ring threading pumps can operate across vesicle or cell membranes. And that's still unknown. No one has reported on that. Of course, just to remind you, many of nature's molecular pumps work across cell membranes, like the sodium-potassium pump, and that is really essential for their function. So that's a very exciting question. I also think that whether artificial molecular pumps can transport rings across membranes could have some pretty big medical potential as a kind of therapeutic, an alternative therapeutic agent. So there are plenty of macrocycles out there that have therapeutic effect, including oligopeptides like cyclosporine and also beta-cyclodextrin, which is our kind of classic ring in supramolecular chemistry. And so if we were able to show that artificial molecular pumps could transport these rings across membranes, I think that would really open up this technology for medical application to see whether by selectively pumping these rings across cell membranes, you could achieve some kind of therapeutic effect. And that is the end of the talk. Thank you for listening, and I'll take any questions.
Amazing. Thank you so much. Really appreciate it. I would love to dig into a little bit more of your future projections here, but maybe we'll just start with a few questions from the audience for now. And I'll just go line by line. There's already a few. If you're in the audience and want to ask a question, you can still do so by putting it in the chat or just raising your hand, but I will start with the questions in the chat. And so we have Paritosh first. Do you want to unmute and ask the question yourself, or do you want me to ask it?
Hey, thank you so much for the talk. I'm just curious, why are the natural pumps heavier and what's the reasoning behind their heavy weight?
What's the reasoning behind their what, sorry?
Like their heaviness, like why are they heavy? 10x almost. Yeah.
Sure. Yeah, I can give some answers to that, I think. Let me go back to that slide. I was comparing those pumps. They're heavy relative to ring threading pumps, right? But they accomplish a lot more than those ring threading pumps because so those natural molecular pumps, they have to embed themselves in the cell membranes. So that is one reason that they have to have a particular kind of structure. They have to have lots of transmembrane domains in order to achieve that. They also have to embed themselves in cell membranes with the correct directionality, right? The pump's worse than useless if it is pointing the wrong way. Whereas these ring threading molecular pumps, they're just operated in free solution or attached to other large aggregates directly. So that is maybe one reason. Also, if we think about how those natural molecular pumps transport solutes, they do it by a different mechanism that I guess inherently requires more atoms to achieve. So in the case of ring threading pumps, the cargo actually threads the pump, or the pump goes through the cargo. In the case of the natural molecular pumps, it's the opposite scenario. The natural molecular pump has to create a space in the hydrophobic membrane through which the solute can pass. And generally these solutes, like ions, do not want to spend much time in hydrophobic membranes. And so it has to create an entire passage across the length of this transmembrane domain. And so to achieve that, it has to have this at least some kind of barrel shape temporarily, which is going to take a lot of atoms to achieve. So those are the best two reasons I can give why natural pumps are bigger.
Happy? For now. Okay. Feel free to ask a follow-up in the chat. Okay, you've got a thanks. Awesome. Next on up, we have Stefan Bosny. You have quite a few questions. Do you want to unmute and ask them yourself? Otherwise, I'm happy to read them out.
Yeah, I'll go for it. Hi, Jim. Cheers for the talk. So yeah, what happens if you try and do multiple cycles of pumping? Do you eventually get too much charge in there and disrupt the micelles? And are you specific for the different size of micelle? Could you use that as a cool like disruption mechanism for measuring how much pumping you've done or something?
I think it probably would destroy the micelles eventually. I did perform a second cycle, but I didn't do any TEM imaging those, so I can't say for certain, what the morphology is. There is also the possibility that the morphology of the micelles is going to change before they are completely destroyed or separated into unimers. Yeah, just for this study, I opted to make the comparison to their small molecule pumps rather than to go down the road of trying to destroy the micelles, basically because it would just be a ton of work to pump every cycle and then measure all the results afterwards. But I think that you're right. I think it would result in destruction, and also that it would depend on the length of the polymer chain, because pluronics larger pleuronics, and especially pleuronics with larger PPG segments, are much more readily form micelles. And so to overcome that, I guess you would need more rings.
Yeah. And so I guess related to that, do you have a feel for whether your macrocycles in the oxidized form, if they're sitting on the ethylene section or the propylene section of the polymer?
I can't say for certain, actually. See, you can distinguish between different parts of the chain with dosing NMR. But the thing about these macrocycles or these polyrotaxanes is that, as far as we know, the rings are mobile. And so they, shuttle along the entire length of the chains. And so they have their own translational diffusion even within the micellar diffusion. So dosing NMR wasn't really able to speak to that question, as far as I know. And apart from that, by proton NMR or other techniques, I couldn't really tell for certain. I would guess, though, that they would prefer to reside on the peg, right? It being more hydrophilic.
Okay, great. Thanks.
Thanks.
Cool. Now, the next one, we have Emanuele.
Yeah, nice talk, Jim. I just want to ask you if you have any idea or view of why pumping in micelles is less efficient. Is it just a matter of crowding of the pumping units, or is there something else?
I think I missed a key part of that question. Can you say
sorry.
again?
So the question is why, if you have any idea of why pumping in micelles is less efficient than pumping in non-micelles. What's your thoughts about that?
The main piece of evidence I had on that question was the UV vis. And that showed that there were some competitive Viologen interactions in the micellar scenario that were not present in the small molecule control study. And so I think you maybe alluded to this the first time you asked the question, that by positioning these molecular pumps in close proximity, we're probably, prearranging them to have some kind of competitive Viologen to Viologen interaction. And it could be that interaction just directly competes with formation of the tris radical tricationic complex, which is necessary for pumping. But just based on those UV vis results, I can't say for certain whether it is the BIPs or the Viologen units in the pumps themselves, or possibly it could involve the Viologens in the rings that are forming that competitive interaction.
Good. All right. Let me know if you have a follow-up here, Emanuele. Otherwise, I'll go to Steve Vedder.
No, it's good. Thanks.
Cool. Thank you.
Hi, thanks for your work and for sharing it with us. I was wondering what the energy source was for your pump. Is it just a matter of changing the pH of the solution, and is there any work towards, you were talking about putting it into a actual cell at some point. Are you already starting work at using ATP?
So the driving force for these pumps is redox chemistry. So I add zinc powder to the solution and it donates a bunch of electrons to the system. That's how I form the key host gas complex. That's the first stage of pumping. And then oxidation using oxygen in air to remove those electrons again, return the pump to its ground state. So in effect, I'm using chemical energy in the form of solid zinc and then gaseous or dissolved oxygen.
You're reversing entropy. You're reversing entropy externally, basically.
I would say it's driven maybe by the enthalpy of the host gas complex. Yeah.
All right.
But yeah, sorry. There was another part to Steve's question that was the pumping into vesicles or cell membranes. So I'm not doing that. I'm actually graduating in 2 days. So I am done with these molecular pumps for now. But I am sure that there must be people, because as I mentioned, this is just one example of a ring threading artificial molecular pump. There are a bunch out there in the literature. And I'm sure that other research groups must be trying something like this. Whether it would work by ATP, I think that would be maybe another project into itself to try and get it driven by catalysis of ATP. probably for now, the most likely thing is people would use their abiotic pumping mechanism to try and achieve it in vesicles or cell membranes.
All right. Sounds. There's no follow-up. Let me know. Jeremy. Wow, we're breathing through the questions here. I love it. Jeremy, you go. Otherwise, I can read out his question. How much chemical potential can these systems generate? I'm concerned about moving these hydrophilic rings through cell membranes. Overcoming the hydrophobic barrier in the cell membrane seems tricky. Biology tends to move to an internal channel to minimize exposure to the membrane hydrophobicity. Wow, that's quite the tongue breaker here.
Yeah, I think that's a good point. As to the first part was chemical potential. Actually, the energy of stored rings or the chemical potential of the stored rings has not been measured by us. And I don't think for any ring threading pump, it has been directly measured. I think that's actually quite challenging to do, especially for small molecule rotaxanes. There was one paper from the Stoddart group in 2020 where they attached a photocleavable group to the end of the chain that the ring was stored on. And so, by cleaving that stopper and then releasing the ring back to solution, you could measure the rate of release of the ring. And from that, you could infer the energy of the release of the ring. So that is one way by which it might be possible. In that particular instance, the ring release was just too fast, and it was basically limited by the release of the stopper. The sort of innate energy of the rings couldn't be calculated. But maybe by a similar kind of experiment that could be done. So that's still unknown, chemical potential. As to transfer through hydrophobic membranes, that's definitely true. And as was mentioned, the natural molecular pumps create a special compartment to allow the transfer of these extremely hydrophilic solutes. I think it's an unknown question. I think really just people have to try it and see what's possible. The rings that I used in this study are extremely hydrophilic. They're tetracationic salts. So you can imagine that trying to pump them through a hydrophobic membrane would be more difficult than trying to pump maybe something like beta-cyclodextrin, which is not charged and which has some hydrophobic character to it. Discounting cell membranes for now, and maybe thinking about polymeric vesicles. So those vesicle walls can be very large. They can be the size of, for example, the pleuronic chains that I talked about. So long polymers of 5,000, 10,000 grams per mole. And so maybe across a transmembrane domain that was a large polymer, it would be more feasible to transport these rings than through the relatively small cell membrane. So that might be a first step to try in vesicles.
All right, wonderful. We have a question from Steve that's also mimicking one that I want to ask, which is, first of all, just a pre-congratulations on your impending graduation. And then a question of what's next for you. You already mentioned, okay, you might be moving onward from this work. Give a little bit of an outlook into what you're going to be focused on next.
I want to stay in the molecular pumps game, artificial molecular pumps. I think it's a really exciting technology with a lot of potential, but I want to learn from the protein side of things. So right now I'm applying for postdocs to be a postdoctoral scholar in protein engineering groups. And hopefully if one of them comes through, I will try my hand at making protein-based versions of artificial molecular pumps. So I feel like now I've learned from the purely artificial side in Fraser's group, and it would be nice to learn from the more proteinaceous natural side.
Wow, that's amazing. Is there a lot of interdisciplinary collaboration happening usually, or do people move from one into the other a lot, or is that relatively new in the pump space?
I would say that there is not a lot of transfer, and I would say that there's also not much communication between people who study natural transporters and people who study these artificial ring threading ones. Yeah, so that's part of the reason I want to give it a go, actually.
Amazing. We wish you the best of luck with it, really. And in terms of next steps, if there's anything that you want to share with folks of how people can stay up to date on your work, that would be super useful. And, any kind of hopes and dreams that you have for the field or encouragements for others to try out. Like basically any last words that you want to share. Now's a great time.
Well, yeah, thanks very much, Alison, for giving me the chance and supporting me. I would say that the future of the field, I think, summed up, to me at least, in these 2 slides. I think these are the most interesting questions for ring threading pumps. Yeah, so I'm excited to see if someone one day is going to pump across a vesicle or a cell membrane, and I'm sure it's in the offing.
We will hope to hear from them if they do. Thank you so much. And early congratulations. I guess in Germany, we don't do early congratulations. Here we go. Take them once you have actually graduated. And yeah, thank you so much for coming today. Thanks everyone for your amazing questions. I really appreciate it. And yeah, I'm very excited to hear from you again soon. Have a good one, everyone.